Okay, a pleasant morning, everyone. By the way, we are going to discuss this morning about enzyme, this will be the topic after the midterm exam. Okay, so what are enzyme? Enzyme, by the way, is different from a catalyst, which is usually used in the laboratory or in inorganic or organic chemistry, because if you talk about catalyst, this is the one which is add on, no? Especially in the laboratory to hasten up or to fasten, no? To hasten up or to fast track the reaction, especially in the laboratory. But if you talk about enzyme, they have the same, no? It will have this will act as a catalyst in a way, but this catalyst will be in the biological catalyst, meaning it is being in the body or in the body system. So this is what we are going to discuss this morning, because this is a very large or big topic. So we'll chop we'll have it into two sessions. So let us start. The outline of this topic is, let us start first with the classification of enzyme and how are you going to name an enzyme? What are the terminologies which are being used in an enzyme? So, and what are the factors that influences the activity of enzyme? What are its mechanism or the action, and how it is being regulated, and the use of enzyme in medicine, and also what is this transition state, okay, of an enzyme. These are the things that we are going to discuss for this topic. Okay, let us start with an enzyme. So the drawing here is where it will act, where the enzyme will act as a catalyst, is which is being have it in that color, that white color there. So what are enzymes? Enzymes by nature are protein in nature, and these enzymes are either in the tertiary or the quaternary structure. So that is an enzyme. They are biological catalysts. So they hasten up, they hasten up every reaction in the body. This will not happen. Most of the reaction in the body will not happen without the presence of catalyst. For instance, the food intake that we have. Okay, so once we eat, what will happen? There are so many enzymes which is needed in order to convert that food that we intake into nutrients. Okay, so the first one is, once we start with the food in our mouth, what will happen? This will be mysticated by salivary amylase. Pagkalisod siguro makaon o dili siya ma-mysticate. Mysticate ka na medyo. Lisod itulon og ang atong pagkaon, wala siya'y salivary enzyme. The salivary enzyme is the one that will, this will make the food that we eat easy to swallow, and that is salivary enzyme. Okay, then after that, different enzymes will be needed in order that will be converted into nutrients that will be needed in the different parts of our body. So that is an enzyme. Without an enzyme, what will happen is it will take too long. The food that we have, it will have a maximum of 4 hours. That is 4 hours for the food to be digested fully and will be used as energy in our body. So that is 4 hours. Pero na uban paspas o metabolism na ay 3 hours or 2 hours lang. But as you grow older, what will happen? Your metabolism will be a little bit slower. That is why mga batanon pa, kusog mga on, pero dili mo tambok. Why? Because their metabolic process is very fast. Kanuman, pas-pas po ang working sa ilang enzyme in their body. But as you grow older, what will happen? Okay, gamay na lang intake of food, but what happened is dugay siya ma-digest. Okay, so that's it. So that is an enzyme. No? So it is protein in nature. The second characteristic of an enzyme is they are catalysts. What do you mean by catalyst? They accelerate a reaction. No? Accelerate meaning ipaspas niya. You know what? When a snake eats a food, no? Iya po na, i-masticate po na niya. Pero what happened is, dugay siya ma-digest. So once a snake will get his prey, what will happen? I-masticate niya. Pag masticate, i-swallow na niya. No? Wala siya mga ngipon. Okay. Pag swallow niya, okay, it will take how many days before it will totally be digested? Ana na siya. Pero ang tao, dali ra. No? So, why? Because of the catalyst that we have in our body. So, your catalyst will not permanently change in the process. Meaning it will not, okay, this will not even in the laboratory. So what we add is a catalyst. For instance, for the boiling system, what we had added is metals sometimes. No? Kanina metals, this will not be converted into such thing, meaning it will not interact with the reactant. Instead, this will only hasten up the reaction. Then afterwards, pwede na po na siya maggamit ng otro. Okay, that is a catalyst, an enzyme. So they are not permanently changed and they will not react. Okay, they will not have a product, this enzyme, because they are only there to be used up in order to speed up the reaction. So that's a catalyst, an enzyme. So if you look into this one, so this site here is known as the enzyme site. This is where your enzyme will work out. So an enzyme is effective. Enzymes are extremely effective. In what way? They will increase the reaction rate. Tanawa ka unsa ka kusog. 10 to the 9, 10 to raise to the 9 power, or 10 to the 20, raise to the 20 times as fast. So that is the importance of an enzyme. So it is extremely effective. So in every process of our body, there is always an enzyme. Okay. So, but what happened is they are extremely specific, meaning, for instance, for salivary amylase, ganyan rin na siya dapita. Pag naog ana, lain na po na nga enzyme. Meaning, dili pwede ang enzyme mo function to different parts of the body. No, there are very specific. Meaning, if that is the substrate, kana ra pod ang iyang, if that is the type of reaction which is needed, kana ra po nga enzyme ang kinahanglan. Okay, that is what is meant by extremely. So first is they are extremely effective. In what way? It will, how many times more it will hasten up the reaction. The second one is extremely specific, meaning very choosy siya. It will catalyze to only certain reaction. Okay. And reusable. As I have said, this will not go with the process. So you can still use it. This will not interact with the process. Okay. So that is the function of an enzyme. Extremely effective, extremely specific, and reusable. Okay. How are enzyme named? Okay. So the reaction here, it could either be, there are so many reactions which is needed with the presence of an enzyme. So from reaction that they catalyze or the compound, the type of the compound, on what they act, there are two ways in naming it. Some of the name is older name na. For instance, the trypsin. Okay. Trypsin is kani. Trypsin is an older name for a catalyst. But in most cases, ang bag-o karon, it is with ending with ase. So, for instance, the sucrase. Sucrase is the catalyst which is used to convert sugar into sucrose. Then, lactase is the catalyst in order to convert that into lactose into sugar into lactose. That is lactase. Same thing with maltase. So sucrase from the word sucrose, conversion into sucrose. What is that catalyst? It is sucrase. Conversion into lactose, it is lactase, then maltase. For the chymotrypsin protein conversion is the protease. So as you can see here, it ends with ASE. In most cases, we are no longer using the older one. Now, we are using the new name of the enzyme ending with ASE. And it's easy to predict what kind of reaction is being used up by this enzyme. For instance, sucrase, meaning conversion into sucrose. Then conversion of lactose, lactase. Conversion of maltose, maltase. So it's easy. Take a look. It's very specific. Ang klase nga enzyme to convert into sucrose, sucrase agad siya. Meaning you could not make use of the enzyme lactase to convert into sucrose. Dili. Ang lactose could only be used by means of lactase. That is it. So that is what is meant by specific. So there are 6 major classification of enzyme. The first one that is the oxireductases. What do you mean by oxireductases? This is the oxidation, catalysis of oxidation and reduction. You have already come across with this when we have the reactions in organic chemistry. Yeah, the oxidation and the reduction process. Okay. The second one is from the word. So if it is this first one, oxidation reaction, so yun ra. I-combine la ang reduction oxidation. That is why it's so called oxido from the word process is oxidation. Reduction, reductases. So, giusa na siya nga classification. Basta gani, the type of reaction is the catalysis of oxidation and reduction. It is oxidoreductases. But if it is the transfer of group of atoms from the word transfer, what kind of an enzyme is used? It is transferases. Okay. Then, if and when hydrolysis reaction, meaning if it is being added with water, catalysis with addition of water, what kind of an enzyme is that? That is hydrolysis. This is a bigger classification of enzyme. Then, if it will catalyze the addition of 2, kani, usara na, kani, this one is, kani transfer lang ni siya, kani is addition gid siya. This is different. The transferases from lyases, transferases is the transfer. Here is the addition. Addition of 2 groups to a double bond, or the removal, either addition or removal of 2 groups, 2 adjacent atoms to create double bond. It is the lyases. That's it. Then, if there is isomerization reaction, what kind of enzyme is used? That is the isomerases. And the last one is, if you're going to join 2 molecules together, what kind of enzyme is used, it is the ligases. So in the incoming PowerPoint, you will see the different example of this 6 classification of an enzyme. So here, no, that is, for instance, no, we have the enzyme. This is now our enzyme, no, the what we call this, the specific enzyme. The major, no, the major classification of enzyme, for instance, is oxireductases. No? Going back to here, what is the oxidoreductases? That is the catalysis using oxidation and reduction. No? So what kind of an enzyme? The lactate dehydrogenase. Take a look. ACE na ni siya. Okay. What do you mean by lactate dehydrogenase? Okay. Meaning this is now your conversion of your lactate into pyruvate. Okay. What kind of enzyme is used? It is the lactate dehydrogenase. Okay. Dehydrogenase meaning you remove hydrogen. Diba? Now take a look. Dehydrogenase. So from lactate with OH here, finally, it removes the hydrogen, no, to form into water here. That what will happen here? The hydrogen and another hydrogen here. No? Okay. You have here, you will form H2 here. So that is known as dehydrogenase, meaning the removal of hydrogen. Okay. What kind of classification? That is oxireductases. The second one is the transferases. Tanaw na to. Let us look into it, what is being transferred. So, as you can see here, diba? There is a transfer of group of atoms that is for the transferases. Okay. So, what is this? What kind of, no? What kind of enzyme is used? That is the aspartate aminotransferase or aspartate aminotransferase. What do you mean by that? Meaning the amino group is being transferred from this word, or aspartate transaminase. Na? So this is your specific enzyme, but the major classification of this is the transferases. Kaya ba nga naman, it transfers your amino. Where is your amino here in your aspartate? Here, this is the amino. No? Your amino and the aspartate and the alpha ketoglutarate, what happened? Your amino here will transfer to your alpha glutarate to, no, convert that into the glutamate. And your aspartate will be converted into oxaloacetate. Nga naman, because there is already a transfer of amino group in your alpha glutarate and converted that into glutamate. Kadumdum mo ani, this is the what? The formula of glutamic acid. Okay, here. That's it. What kind of enzyme here? It is the transferases. Nga naman ang amino nimo gi-transfer. No? Unsa nga klase nga enzyme ang gigamit? The aspartate aminotransferase or aspartate transaminase. The third one, the third classification is the hydrolases. Okay, from the word hydrolases, meaning what? Your water is being, this is it. Your water is being used up. That is hydrolases. What kind of an enzyme? That is the acetylcholinesterase. Okay. There is an ester here. Okay. Here, this is now your ester. So this is the acetylcholine, okay, with the presence of added with water. what happened? Na-disintegrate ni siya. Na-disintegrate into acetic acid and the choline. Okay. By means of cholinesterase enzyme. Then, the major classification of this is hydrolysis, meaning it is added with water. Once it is added with water, this will converted into two compound, that is the acetic acid and the choline. Okay, that is for the third. Another one is the lysis. What is lysis, by the way? Here, lysis is the addition of two groups to a double bond, or the removal of two groups from adjacent. So it is either the addition or the removal. Basta duha ka grupo. Okay? Here it is. So take a look. This is lysis. What kind of an enzyme is used? The aconitase. Okay. So there is an addition of water. Once you have this, what will happen? This will, from cis aconitate, this will be converted into isocitrate. In what way? So this is OH, di ba? HOH. So what happened? Canisia is being add on to this kani. Two groups. Kani gibutangan hiya. Then where is your other hydrogen? Your other hydrogen is added to this. Your OH, this HOH, di ba? The OH here is added to this carbon, and the H is added to this carbon. Okay. So two groups are being added, and that is lysis. The fifth class of enzyme is the isomerases. An example for this is the phosphohexose isomerase. What is isomerization? Isomerization is just, what happened in the isomerization? Ang cleaving niya, ang iyang cleaving niya is ma-disarrange lang. Take a look. So here we have the glucose 6-phosphate. Kung sa may nahitabo, kaning glucose 6-phosphate, it becomes into fructose 6-phosphate. What kind of enzyme is used? That is the phosphohexose isomerase. So there is only in the isomerization, what happened? There is a disintegration or a change in the bond. Okay. So from glucose 6-phosphate, it becomes into fructose 6-phosphate. And the last one is ligases. What is ligases? That is here, kani addition of two groups from the double bond to the double bond or removal. Kani is joining two molecules na. Kani addition of two groups from the double bond. Okay. Here, what happened? Joining of two molecules na. So for instance, we have ligases. The last one. We have what kind of enzyme is it? Tyrosine tRNA synthesis. Okay, so what happened? There will be addition of two groups. We have the ATP plus the tyrosine, the transfer RNA. Okay, so you come up now with addition of two groups. Kaniya, lahi na. This is the adenosine monophosphate and the phosphate inorganic phosphate here. Okay, so that is for the ligases. So how do enzyme work? Actually, tungod sa kadako sa energy activation, dili siya ka-form of product. So the function of an enzyme, no, this will lower the energy of activation. This yellow here, no, this yellow here, no, is the activation energy without an enzyme. Di ba taas kayo? No? Taas kayo. Kaning pula here is the activation energy with an enzyme. No? So, kung i-lower, no? Kung i-lower sa enzyme ang activation energy, what will happen? No? Substrate energy level, so there will be a net energy that will be released in the reaction. So what will happen? Reaction will push through into the product. So here, what happens lies in fact that most chemical reaction that release energy, which is exothermic, outside, requires an input, kay gawas man siya. So kinahanglan siya og input inside, so that there will be a product that will be formed. How? Your enzyme will lower its activation energy. Kaning pula, mao na ni siya. Kaning pula is the energy wherein is needed in order to come up with a product. So requires input of some energy to get them going. So the initial input of energy is called the activation energy. So kinahanglan siya activation energy, pero tungod ka sa kadako sa activation energy nga ni, kay walay enzyme, this will not produce a product. Unlike with this energy activation here with an enzyme, this will release the reaction into a product. So enzyme work by weakening the bonds, which lowers the activation energy. So ang mga bond ngadto sa imong mga reactant, what will happen? This will loosen up, okay, in order to lower the lower its activation energy. So enzyme lowers activation energy to catalyze the reaction in order to come up with a product. So for instance here, you have this test tube here is enzyme without substrate. The substrate that we use is the hydrogen peroxide. Okay. Later on we'll discuss what is substrate. Okay. So here without enzyme without substrate, there will be no reaction. Wala. Wa kay makitaan nga bubbles or whatever. No reaction. Here, this test tube here is enzyme plus substrate, meaning we have hydrogen peroxide. So what happened? You form, you can see here bubbles, kani, Why? Because there is already a reaction. So your enzyme reacts with your substrate. Kina naman siya substrate. Kani without substrate, no reaction. Kani with substrate, na ay reaction. And the other one, this one, we have a liver homogenate, catalase. Okay, so we have a catalase. That catalase is the catalase enzyme. As you can see here, there is already a reaction. Okay, so what does it mean by this? It needs two, in order to come up with a reaction. It needs an enzyme. It needs the presence also of a substrate. Enzyme plus substrate, it will form a reaction. So let us introduce the common terminology which is used here in an enzyme. The first one is what are cofactors. So we already know that enzymes are protein in nature, diba? But there are enzymes that contains non-protein portions and they are known as cofactors. What are apoenzymes? These are protein polypeptide portion of an enzyme. So an enzyme with a polypeptide, meaning dako-dako siya na protein. It's a polypeptide portion is an apoenzyme. What What are coenzyme? These are organic cofactors. What are cofactors? Cofactors are non-protein portion of an enzyme. So if it is organic, na, it is a coenzyme. Okay. For example, vitamin B. Kaning vitamin B is sometimes it is being into 3, that is 1, 6, 12. B1, B6, B12. Mo nang ginatawag og multivitamins, vitamin B. Na? So, what is the importance of this vitamin B, especially sa mga idaran na? They are essentials to the activity of enzymes. So, if you intake, as I have said, mga older people, their metabolic process, their enzyme activity or their metabolic activities are a little bit slower na. So we need this vitamin 1,6,12 nga naman. This will activate. This will help in many enzyme activity. That is why mag 40s na gani, mga nanay, tatay ninyo, barato naman yung vitamin B1612. So what will happen? Ang ilang metabolic process, ang enzyme, makatabang ni siya. Na? So, barato na ni siya. Naitag 3 pesos, naitag 5 pesos. 1,612 tawag ana. You let the older people drink this with vitamin C also. Okay. Ako na pa vitamin E, Myra E. Aron ang mga kunot-kunot, mga kuan, char. Ayaw sa mong vitamin E, kaya nga naman, bata pa man mo. So, it's easy to replenish the dead tissues, dali ra kayo sa inyo. But for the older people, lisod na siya. So that is why most of the older people needs to take more vitamins. I'm taking 3, the list. That is vitamin 1,612, B1612, vitamin C. Pero kamo kinahanglan mo vitamin C. Kinahanglan na siya. Vitamin C is needed in order for preventing this will activate your, for instance, for diseases, okay, aron layo-layo sa mga sakit-sakit. That is vitamin C. And vitamin E for older people, aron dali matibuwang. Wow. Pero mahal ang vitamin E. That is, for example of that is Myra E, tag 14. But it's nice to drink these vitamins for a longer, beautiful life. Char. Okay. Now, an example of coenzyme, that is vitamin B, heme also, part of hemoglobin, and heme is a part of hemoglobin. In the blood clotting, yeah, that is heme. Part hemoglobin and several oxireductase. Okay. So, what are substrate? Okay. Substrate are, these are the reactant, reactant, or an enzyme that acts on is the substrate. So where your enzyme acts on, asa siya activity niya buhaton is known as the substrate. Okay. So if this is an enzyme, then this enzyme needs to act on the substrate. Okay, that's the substrate there. This is the enzyme. This is the substrate. Okay, so this will join. Join. Your enzyme will act on your substrate. It will join to have an enzyme substrate complex. I repeat, when enzyme joins your substrate or act on your substrate, it will be, what is that activity? It is the enzyme substrate complex. Unsa ang substrate? Maong diha hing act or hing ang inyong enzyme nagtrabaho. Aron iyang i-convert into something else, into a product. Aron ma-hasten up ang pag-convert into a product, and that is enzyme substrate complex. Okay, what is active site? Yung bi ako choosy ni enzyme. Okay, it's choosy. Okay, so enzyme will only act on the active site. What is active site? Active site is a restricted region of an enzyme molecule which binds to the substrate. So if you have the substrate, naagyo na siya place nga ang enzyme nganha ra gyud makalihok, and that is known as the active site. So if this is this enzyme, no, the active site is nganha kani naadapit ang active site. So nganha mo sulod ang imong enzyme sa active site. Okay. Na? So, what are these different enzyme action? Okay. So, if you look into the first drawing, this is your substrate. Na? This is your active site. No? Illustrate na siya nga kinahanglan mo sulod, mo conform. No? Kinahanglan pareho og shape. Okay, take a look. No? So, this is your substrate. This is your active site. Unsa man ni siya? Kani unsa ni siya? Mao ni imong enzyme, no? Kani ang imong enzyme mao ang imong substrate. Ang active site kani mao ni active site kani. Okay. So kung muhaom gani siya sa active site, this is substrate entering the active site of an enzyme. Unsa ni siya? This is your enzyme, this is your substrate. Katong iyang hauman, na, the one that will where your enzyme substrate will interact is known as the active site. So enzyme changes shape slightly as substrate binds. So if muhaom siya, no, meaning your enzyme and your substrate binds together. So this is known as, katong gisulis ganina, enzyme substrate complex. What is enzyme substrate complex? When your enzyme acts with your substrate. Okay. So you have now, your product will be enzyme product complex. Okay. Enzyme product complex kani siya. And finally, mo produce na kag product. Tapos kani imong enzyme will leave. No, will leave the active site. Your product will leave the active site, pero maura gyapon enzyme nimo. Then kaning enzyme, pwede na siya balik na po gani. Okay. As I have said, because this is not used up. It is reusable. Your enzyme is reusable if and when you have already a product. And this is known as enzyme action. So what is important in the enzyme action is it will fit in. The enzyme will fit in, the substrate will fit into the active site which is present in your enzyme. Once it will fit in, it will have, what is the name of that? That is enzyme substrate complex. And when it will react, what will happen? That is enzyme product complex. And once you will produce a product, it will leave the active site and mag-separate na dayon ang product o ang enzyme. Then it goes back. Your enzyme will go back here like that. So that is the enzyme action. What is induce? Induce fit or induce fit. Okay. As you can see here, slightly mo conform siya sa shape. Mo fit in siya sa shape. Okay. So that is what is meant by induce fit. I change the shape of the enzyme, shape of an enzyme active site. Okay. Then induced by the substrate. It is the substrate that will slightly change its shape to conform with the active site of your enzyme. Okay, take a look here. So this is your enzyme. So ang imong ang imong substrate kinala mo conform siya sa shape ani, no? That is it. So in the induced fit, what happened? A change in the configuration of an enzyme active site. Okay, that is, for instance, the hydrogen and the ionic bonds are involved. No, the hydrogen and ionic bonds. What are ionic bonds? There is a transfer. Diba yung transfer siya? Okay. So in the induced, it is the substrate that will fit in to your enzyme. Okay. So this is the active site. This, the whole is the enzyme. This is your active site. Kanina ni active site. So kinahanglan mo fit in ang imong substrate. Okay. Induce. Kay kabalo ba mo og kanang mga nakay? Nay mga nakay nga dugay gyud, mga anak. Oh. Most of those who doesn't want to undergo CS, cesarean section, what happened is they will be kanang dugay magbati. They called it as they add on. What happened is it will be injected with inducer. Okay, it's a medicine wherein this will induce, pero sakit siya kay ipapaspas ang what they call this labor. Na papaspas ang labor. So what happened is that is what is meant by induce. Instead of it will take 3 days nga nag-labor, but if you are going to inject it with inducer, what will happen? Within a day, manganak na siya. Pero ang labor is sakit kay paspason man niya ang pag-labor. No? That is an induced kind of labor. Here, what happened? This induce is the substrate will fit in, will find ways in order to fit into the active site. Okay. So, what will affect enzyme activity? There are 3 factors that affect enzyme activity. First is the environmental conditions. What do you mean by environmental conditions? Second is cofactors and coenzyme. And the third one is enzyme inhibitors. Let us take it one at a time. Okay. In the environmental condition, one of it is the temperature. No? Extreme, meaning a higher temperature, taas ra ang temperature, what will happen? This will not happen. Okay, tendency ang imong enzyme ma-weaken or mawala. Na? So high temperature may denature or unfold the enzyme. Na? Denature meaning it will no longer function as is. For instance, it is for the transferases, but because of higher temperature, ang mga unfolding, what you call this, ang iyang activity will no longer be for transfer of the atoms. And that is one of the environmental condition. That is high temperature. Second is the pH. The pH concentration wherein mostly enzyme will act upon is between pH 6 to 8, meaning 6, not slightly, that is 6 is slightly acidic, then 8 is slightly basic. 6 to 8. So it could be slightly acidic and slightly basic or near neutral. What's neutral? It's 7. That is the pH. Or else, ma-denature gihapon ang enzyme ninyo. No? Meaning its function will no longer be the same as an enzyme. Then, the ionic concentration. Ionic concentration is if it is salty, the salt ions. So those are environmental conditions. The second one is the cofactors and the coenzyme. What are cofactors? These are those enzymes which is not a protein in nature. No? Okay. So Cofactors, for instance, inorganic substances, no? Presence of zinc, iron, and vitamins respectively are sometimes needed for enzymatic activity. So kinahanglan gihapon, ito akong sulti sa inyo nga, kinahanglan yun ang vitamins in order to hasten up, no? Or to add on to the strength of your enzyme. Okay. So, need for proper enzymatic activity. Example, iron must be present in the quaternary structure hemoglobin in order to pick up oxygen. So, kinahanglan ng iron, no? In order that your quaternary structure, no? The enzyme in your quaternary structure to come up, no? Into the picking of hemoglobin, no? Into by its function of picking up the oxygen. So, kinahanglan niya og iron. No? Less ang iron mo nang that is why we need an intake. No? Mo bito ng anemia. Anemic gani ka meaning you lack iron. So, you need to take vitamins for iron. Okay. Vitamins for iron is very minimal in price. Barato ra siya. Okay. So, what are examples of enzyme inhibitors? The first type of enzyme inhibitor is the competitive inhibitor. From the word competitive, competitive meaning there will be a competition between an enzyme. So competitive inhibitors are chemicals that resemble an enzyme, normal substrate, and compete. Your enzyme, enzyme, what will happen? Your enzyme has an active site. In the competitive inhibitors, naay chemicals nga mo resemble, halos pareho sa normal substrate. Tapos, your substrate, di ba, ang mo bind ngadto sa imong, mo bind sa imong active site if this is now your, for instance, this is your active site. Okay, this is your enzyme. Okay, so it needs a substrate that will fit into this. Okay, so naay substrate, pero naay chemicals nga mo resemble sa imong substrate. No? So duha na sila. That is why it's so-called competitive inhibitor. Your substrate nga tinuod ug ang imong hing, na? That's the normal substrate. The other one is not the normal substrate. It is a chemicals that resemble to your substrate. So these two will compete in the binding site. And that is what is meant by competitive inhibitor. Take a look here. So this is your active site. Na competitive inhibitor nga niya. Naapong yung kay substrate. So this will compete in the binding in the active site. Kaning competitive inhibitor and the substrate. And that is what is meant by competitive inhibitor. These two will compete in the active site. Ang tinuod nga substrate o ang competitive inhibitor. Your competitive inhibitor are these are just chemicals that resembles your normal substrate. Okay, so example of competitive inhibition, for example, ethanol. So the metabolism of ethanol. So ethanol is metabolized in the body by oxidation to acetaldehyde. Humanataan is organic chemistry, which in turn further oxidized to acetic acid by aldehyde oxidase enzyme. So the one that will convert your acetic acid is known as the oxidase enzyme into aldehyde. Then the second reaction is this will be converted into acetaldehyde. Aron dili ni siya mo accumulate sa body. Okay, this is an example. Okay, here. So a drug disulfiram, that is the antabuse, no, inhibits the aldehyde oxidase, which causes the accumulation of acetaldehyde with subsequent unpleasant side effect of nausea and vomiting. This drug is sometimes used to help people overcome the drinking habit. So this drug disulfiram antabuse, tawag ani antabuse, is known as the competitive inhibitor. Na? Kani siya mo inhibit na sa aldehyde oxidase na nga mo accumulate na sa atong lawas. Accumulate by means of acetaldehyde, na. Pero na siya mga unpleasant na side effects kanang magsuka-suka o maglipong-lipong, no. Pero this is very important katong mga na ay not only drug addiction, kung na drug addiction pero na po alcoholic addiction, na. This is a way of controlling those people who have alcoholic addiction, na alcoholic addiction. So paimno ni sila, then they will have no more what you call this, longing to drink alcohol. Nagitaw nga dili pwede nga dili ka inom og 1 shot a day. Okay, nganuman it becomes a habit. In order to eliminate this, it needs a drug named disulfiram. Kaya nga naman, this will inhibit. It is a competitive inhibitor. Na? So, this will inhibit your ways of longing to drink alcohol. Okay, that is an example of competitive inhibition. So, what are its practical uses in medicine? So, for instance, the methanol poisoning. Once this will be taken, what will happen? So a visitor is taken to the emergency room where he is diagnosed with methanol poisoning. Methanol poisoning, yes, because methanol is a wood alcohol, and it's very poisonous once ingested. Dili pareho na siya sa alcohol nga, which is done by means of fermentation sa ethyl alcohol. Okay, kanang mga alcohol nga gina-intake sa mga palahubog. Okay, di kay na siya kusog, di na siya poisonous, unlike with methanol, methanol alcohol, it's a methyl alcohol, not ethyl alcohol. Ethyl alcohol man ang byproduct from sugarcane. Then isopropyl alcohol is the byproduct from the gasoline, like that. So di kay na siya hadlok. Kani maoy hadlok, that methyl alcohol, the methanol. Once ingested, this could poison. So that is methanol poisoning. So what happened is we need a competitive inhibitor katong naka-intake ana. So methanol is metabolized to formaldehyde and formic acid by alcohol dehydrogenase. So advise the patient to get the patient very drunk, since ethanol competes with methanol. So how are we going to cure this? Kung ang tao naka-intake og methanol, kinahanglan pa imnon siya daghang what? Okay. Alcoholic beverages nga naay ethyl alcohol. Nganuman, ang ethyl alcohol will compete, or the ethanol will compete with methanol. This is competitive inhibition. As ang binding site, the binding site, and that will be alcohol dehydrogenase, mao ni enzyme. So mag-compete sila. So it slows the metabolism of methanol. So ang methanol, ang metabolism niya, it will lessen. Instead, it will allow the metabolism of your ethyl alcohol. So this will build up, before they will build up dangerous level. So, mo lower down ang level sa methanol mo compare nimo sa ethanol. And that is competitive inhibition. So to, kung maka-ingest ang taog methanol, unsay tambal, paimnon o hubugon. Hubugon siya o katong naay ethyl alcohol. Basta kay tanduay ba, ana, basta all of that. No? Basta ethyl ang content. Kaya nga naman, this will compete with the binding site, the alcohol dehydrogenase. Kani ang binding site. This is the end site. And what will happen? This will slows down the metabolism of methanol. Allowing toxic metabolites to be disposed. So, iyarang kay wa man magamit, di ba? Kay nga naman hingkumpit man ang inyong ethanol sa methanol, so wala magamit tong methanol, wala siya mo build up sa inyong system, instead gigawas sa inyong system. And that is example of competitive inhibition. So what are inhibitors? There are two types of inhibitors, the competitive and the non-competitive. Okay. What are non-competitive inhibitors? These are inhibitors that do not enter the active site. Wala, di siya kasulod sa active site, but bind to another part of the enzyme, causing the enzyme to change its shape, which in turn alters the active site. Maldito ning non-competitive inhibitors. So, inhibitors that do not enter. Dili siya sa active site mo enter. Instead, gubaon niya ang shape sa imong enzyme. Ug mahitabo, ma-alter ang shape sa active site. And that is non-competitive inhibitor. So, example for this, if this is your enzyme, okay, this is, okay, kana may active site. Yana, okay. Iyang gubaon. Here is your non-competitive inhibitor. Kani, this is your non-competitive inhibitor. So, what will happen is this will change the shape of your active site. So, ang first is competitive inhibitor, and the second one is non-competitive inhibitor. What are competitive inhibitor? This will compete. These are non-competitive inhibitor are almost almost take a look here. What happened in the competitive inhibitor? Competitive inhibitor, it will resemble the substrate, and this will compete in the binding site. This is your substrate, this is your competitive inhibitor, this is the active site. This will compete in the binding of the active site. Makig-competensya sa substrate pagsulod sa active site. Pero lahi ang non-competitive. Kung sa ang non-competitive, maghimo siya. Di siya makig-compete sa active site. Instead, adto siya sa enzyme, tapos gubaon niya ang shape sa enzyme. Pag guba niya shape sa enzyme, what will happen? Maglisod, gubaon niya. Maglisod na sulod ang substrate. Kaya nga naman, giguba naman niya ang shape sa imong active site. And that is non-competitive inhibitor. Okay, so these ends are part 1 of our enzyme. So, let us move on by the next video for our second part of an enzyme. As a recap, you already know what is an enzyme, what is its function, what are its classification. Example of this classification of an enzyme, there are 6 classification of an enzyme. What are example of this enzyme? No? Classification of an enzyme. Also, we touch about the so-called the activation energy, no? How and when this will happen, okay, by lowering its activation energy. We also touch about inhibitors. Okay, there are two types of inhibitors, the competitive and the non-competitive. We had also introduced terms such as what is active site, what is substrate, what is cofactors, no? What are apoenzymes. So let us have the next video for summing up another subtopics in enzyme. Thank you for listening and keep safe, everyone. Goodbye.